Everybody, my name is Jazlyn Gonzalez, and I am part of group number 6 with Andres Garza. Today, we will be presenting over our metabolic tests. Metabolic tests are used to identify bacteria based on their biochemical activities and nutrient utilization. Each species of bacteria has unique metabolic pathways that allow it to produce, consume, or transform specific compounds. Microbiologists determine how microorganisms obtain energy and carbon and use this information for classification and diagnosis. In this lab, several metabolic tests were performed to analyze bacterial fermentation and oxidation abilities. Oxidation fermentation or OF glucose test, triple sugar iron agar or TSI test, and phenol red carbohydrate broth test help in distinguishing between organisms that metabolize carbohydrates oxidatively or fermentatively, detect hydrogen sulfide and gas production, and identify the ability to ferment sugars such as sucrose, lactose, and glucose. Each media contains indicators that change color depending on the pH, allowing visual detection of metabolic activity. For example, phenol red changes from red to yellow in the presence of acid from carbohydrate fermentation, while OF medium uses bromethyl blue to show acid production under aerobic and anaerobic conditions. TSI detects sugar fermentation, gas formation, and hydrogen sulfide production through color changes and black precipitates. Overall, these types of metabolic tests are essential in confirming species identity and understanding microbial metabolism. For our materials, we used 11 OF glucose medium, 10 TSI-A, and for our carbohydrate broth tubes, we used 6 glucose broth, 5 lactose broth, and 5 sucrose broth. For cultures, we used Alcaligenes faecalis, Pseudomonas aeruginosa, Escherichia coli, Citrobacter frundi, Salmonella typhimurium, Shigella flexneri, Staphylococcus aureus, Proteus mirabilis, and our unknowns number 136 and number 276. For equipment, we used a butane tank as our aseptic heat source, an inoculating loop, inoculating needle, and mineral oil. Now for our procedures. For OF glucose medium, 2 tubes of an oxidative fermentative glucose medium were inoculated using an inoculating needle, stabbing the glucose 1/4 inch from the bottom. For each designated culture of P. aeruginosa, E. coli, A. faecalis, and our unknowns number 136 and number 276. 1 to 2 milliliters of mineral oil, which prevents the diffusion of oxygen into the medium, creating an anaerobic condition in the tube, was then added to the tube labeled closed. The other tube was labeled open and was made sure to not be closed tightly. The tubes were then incubated. For TSIA, an inoculating needle was used to touch the top of a colony, followed by inoculating the TSI tube by stabbing through the center of the medium to the bottom of the tube and then streaked the surface of the agar slat. The cap was left loose. The tubes were then incubated. For carbohydrate broth tubes, a single colony was retrieved using an inoculating loop, followed by inoculation of its designated medium. The tube cap was loose, placed back on, and incubated.
Now for the results and observations of oxidative fermentation for glucose medium. For the results and observations, we're going to start off with P. aeruginosa. For the open, we have no change, and closed, we can see that there's a green presence, which means that it's non-oxidative. And also, just a heads up, for most of these, the results are pretty much gonna remain consistent, for the exception of one of them. As for E. coli, it is starting off with the open, we have no change, and for the closed, we have green color, which means that it's non-oxidative. As for A. facialis, on the open, we have a blue tint, which means that there is no acid detected. So this is the one that it was different compared to the other ones. However, for the closed A. facialis, it was green, which meant that it was non-oxidative as well. For unknown 136, on open, we have no change present, and for closed, it was green, which meant non-oxidative. And lastly, for unknown 276, the open, we had no change. And for closed, it was green, which meant that it was non-oxidative as well.
Now for the results and observations of TSIA. Here on the left is pictured our control. For P. aeruginosa, there seemed to be no color change or growth visible. This indicated that this tube was a non-fermenter of the 3 sugars. For E. coli, there also seemed to be no color change or growth visible, also indicating that it was a non-fermenter of the 3 sugars. For C. frundi, there was visible color change surrounding a colony growth that is visible on the slant of the tube. However, it did not match any results, so the results were deemed inaccurate. For S. diffemerium, the butt of the tube turned yellow, and the slant turned a pinkish-reddish color with visible growth of our culture. This test indicated that it only fermented glucose. S. flexneri, there was no color change or growth visible, also indicating that it was a non-fermenter of all 3 sugars. For S. auris, the butt of the tube turned a lighter shade of orange, and the slant looks to have stayed its original color. This test was deemed a non-fermenter of all 3 sugars. For P. mirabilis, the whole tube turned a lighter shade of orange with visible culture growth along the slant. However, no detectable sugar fermentation was detected, deeming it a non-fermenter. For our unknown number 136, the whole tube turned yellow with slight growth visible along the slant. This indicated that lactose and or sucrose was fermented, or there was fermentation of all 3 sugars. Lastly, for our unknown number 276, there was no visible color change or growth, indicating a non-fermenter.
Moving on to the results and observations for glucose broth. Starting off with the results and observations, we're going to take a quick look at the control group just to understand what's going on. So pretty much the control group maintains a clear appearance all throughout with no bubbles that are present within the durum tube. The durum tube is also just to detect if there's any production of gas and fermentation within the bacterial culture. So we can easily see this for E. coli. There was a presence of an aerogenic bacterium, and we can see this because there is fermentation and gas production due to the visible bubble in the durum tube. As for S. aureus, however, there was only a clear appearance on the top, but a murky presentation at the bottom, which showed that there was a growth of bacterium. For P. aeruginosa, we can see that there's a yellow presentation. However, we do have a mild clarity all throughout the tube. As for unknown 136, we can see that there is a clear appearance on top and a murky presentation at the bottom, which also indicates that there is the presence of bacterial growth. And lastly, for unknown 276, we can see that there is a yellow presentation with mild clarity all throughout. So I also want to address that the reason to why there might be mild clarity all throughout is probably because we mixed, or we kind of like shake the tube a little bit so that the bacterial growth can be dispersed all throughout, whereas the other ones where there is only a murky presentation at the bottom, those weren't mixed as much. Now for the results and observations of lactose broth. So starting off with understanding what the control group means. So initially it's going to be a color red, and it only turns yellow whenever acid is produced, which is essentially our pH indicator. So we can easily see this in all 4 of them. For E. coli, there is a yellow color change, which means that acid is produced, indicating that there is a positive test for lactose fermentation, and it pretty much maintains consistent with all 4 of these tests, which shows that there is bacterial growth for each one of them.
Now for the results and observations of our sucrose broth. To the left, you can see our control group. For our E. coli, there was a yellow color change throughout the tube, indicating a positive test, sucrose being fermented, and acid being produced. For P. aeruginosa, there was also a yellow color change throughout the tube, indicating a positive test, sucrose being fermented, and acid being produced. For our unknown number 136, there was a yellow color change throughout the tube, indicating a positive test, sucrose being fermented, and acid being produced. This test was also gas positive due to a bubble inside the Durham tube. Lastly, our unknown number 276 was also shown yellow color change throughout the tube, which indicated a positive test, sucrose fermented, and acid produced.
In conclusion, the metabolic test panel for oxidative fermentation glucose, phenol red carbohydrate broths for glucose, lactose, and sucrose, and TSIA revealed consistent patterns of acid production, gas formation, and hydrogen sulfide that differentiate oxidative and non-fermenters from true fermenters and outline each organism's sugar use profile. By comparing reactions from known controls with the unknown isolate, we used these benchmarks to narrow the unknown's identity based on its biochemical structure. Overall, these results show how routine biochemical testing provides a practical, evidence-based pathway for classifying bacteria before confirmatory and identification methods.
Here's a quick overview of the references used throughout this presentation, from lab notebooks to websites.
So this concludes our video presentation, and thank you guys for listening.